How can we analyze T-cell datasets where we have single-cell measurements of T-cell receptor sequences and the transcriptome? This is Felix Drost from the HELMHOLTZ Center in Munich, and here I present to you mvTCR, a deep learning model for integrating both modalities into a shared representation. Other cells present epitopes bound to the major histocompatibility complex on the cell surface to T cells. These disease-derived peptides can be recognized by the T-cell receptor, which is a highly variable surface protein. Upon this recognition, the T-cell can change its state from a naive to an activated state and initiate a further immune response. When we now want to study the T-cell response, we can take single-cell measurements of the transcriptome, which provides us with information on the cell state. Usually we have this count matrix where for each cell we have the annotation how often a specific gene was detected. And we can use this then for downstream analysis such as visualization, clustering, or differential gene expression analysis. On the other hand, we can have a look on the T-cell receptor as a proxy for what the cell can recognize. Here we can measure the amino acid sequence of a receptor that we can then in turn use as either a barcode to group cells which recognize the same epitope, or we can perform sequence-based analysis here. For example, as you can see here, we can look at the length profile, or we can discover enriched motifs However, both modalities are interlinked. So the T-cell receptor decides what the cell will recognize and thereby the fate of the cell. So it is only intuitive to analyze them jointly. However, if you have a look at the count matrices on the one hand and the amino acid sequences on the other hand, it is not straightforward how we could perform this. That's why we developed mvTCR. That is a deep learning model which is based on a multi-view variational autoencoder and learns a joint representation of both modalities. Per cell, it receives as input the count data for the transcriptome and the CDR3 alpha and beta region representing the T-cell receptor. Here we're using encoding networks, transformer architecture for the T-cell receptor, and multi-layer perceptron for the transcriptome data to obtain a lower dimensional representation of both modalities. This is then in turn fused via the mixture module to a shared representation. This shared representation provides us with a lower dimensional numeric embedding, which captures both the T-cell receptor information on the one hand and the gene expression on the other hand, and can be used for downstream tasks such as clustering and visualization. To train the whole model, we then use decoding networks to reconstruct the original input. We applied this mvTCR on several datasets. First, we had a dataset with cell-level specificity annotation. We found that our joint model, as you can see here in the top row, captures specificity better than a model based on the transcriptome in the second row or in the T-cell receptor in the third row, since it can use the information that is captured in both modalities. Secondly, we applied this model on a large-scale dataset of 750,000 T cells. Here we showed, using the ssci approach of integrating multiple studies, that our model can be easily trained on such large datasets. Using this ssci style integration has the additional benefit that if we want to add novel patients, we don't need to retrain the whole module, but simply adjust for the new patients. Finally, we applied mvTCR to a large-scale dataset in the context of SARS-CoV-2 infection. Here we were able to identify clusters from patients with symptomatic infections that would have otherwise been not present if we simply analyzed on a transcriptomic level alone. These clusters share highly similar T-cell receptors on the one hand and transcriptomic profile in the other hand. And by performing database queries, we further divided them into bystander and disease-specific clusters. To summarize, we developed mvTCR, which is a deep learning model that you can apply to your T-cell datasets, your single-cell datasets, which integrates T-cell receptor information and gene expression information into a shared representation for analysis purpose. In the end, I want to thank everybody involved in this project. And if I now got your interest, drop by at my poster so we can discuss this further, or reach out to me via mail or Twitter.